Hello, my name is Dan Szumski. I'm an independent scholar. I live in Davis, California. The title of my presentation is Introduction to Theoretical Biology. Remember how I began this discussion by categorizing all cellular functions into synthesis and metabolism? So far, we have only dealt with metabolism, or more broadly stated, the cell's interaction with its environment. Now I would like to focus your attention on the cell's synthesis function. Let's review where we are. Our cell consists of a localized region of space that is characterized by reversible thermodynamics and redox chemistry. Its boundary condition is a membrane, which excludes polar molecules and charged ionic species. The respiratory enzyme carbonic anhydrase facilitates transmembrane transport of ions and gases by producing the high-energy intermediary, hydrogen peroxide. This results in a continuous influx of electrons, molecular oxygen, water, nitrogen, and phosphorus across the cell membrane. The stoichiometry of this active transport process is very precise and invariant as long as the cell is normal and viable. The density of electrons within the cell, as measured by intracellular PE, continually rises during the cell cycle, making the cell environment progressively more reduced. We have already seen that this facilitates gas and ion transport and also the cell's adaptability to environmental change. Now we're going to see how that same forcing function, increasing intracellular PE, is also the primary factor propelling cell synthesis forward in time. We will now begin looking at the cell's synthesis. I have not addressed this subject in the same level of detail that you just saw in my discussion of metabolic function. Nevertheless, I want you to see the issues and the opportunities that I see in modeling cell synthesis. Let me begin this discussion by stating the most fundamental law of cell synthesis. Cell synthesis is a least action process where every next step is distinguished from all other possible steps because it is the one that results in the smallest energy change. And as long as the cell remains in this reversible thermodynamic state following that step, the next step is also exactly deterministic, and so on. This makes the synthesis process precisely deterministic, where every next synthesis step is exactly the same as it was the last time the cell's synthesis process was in precisely the same state. This adherence to the physical principles of reversible thermodynamics ensures the high-fidelity replication of the cell's molecular composition. This is what cell synthesis is, after all, what it's all about. You also need to understand that this least action process is the final product of the cell's evolution. This makes its defining characteristics, first, its stability, and second, its precision. This is the cell's script. At some time in the future, it will be possible to catalog the predominant threads in the sequence, and even its many branches. But those details will be unimportant to the present discussion. Here, we are interested in providing context for understanding the cell's most basic synthesis functions. The first element of the synthesis model that needs some context is the continually increasing redox intensity within the cell. In particular, we want to understand, first, how this energy is stored within the cell, and secondly, the way that it contributes to cell function. The cell is a localized region of highly reduced matter. Its reactions occur at an ever-increasing negative PE, or more accurately, an ever-increasing electron activity. I want to suggest to you that increased electron activity doesn't mean that the electron density within the cell has increased. While this is true, it does not define electron activity. Its defining feature is instead the amount of energy stored per electron. And yes, I'm saying that covalent electrons are not in their ground state. Instead, both electrons are in identical excited states, and this is important. It is this elevated energy state that gives rise to electron activity. The identical excited states are a requirement of the reversible process space because its operation must exhibit no energy excess or deficiency. The sum total of the covalent bond's Helmholtz free energy is identically zero. Next, I want you to understand how these excited electron states materialize in our cell model. Remember how, in the discussion of carbonic anhydrase, the oxygen molecule's kinetic energy was quieted to zero. It immediately emerged in the enzyme-substrate complex as a chemical free energy addition to the enzyme-mediated reaction. I want to point out, and this again is important, that it is exactly the opposite of the energy transforms that we see in our world, where free energy goes over into kinetic energy. Here, we see kinetic energy being harvested from the environment and converted to radiation domain energy. More precisely, the energy shows up as an increase in the electron's excited states within covalent bonds. But I want to suggest to you a more profound effect of this energy capture, and that is contribution to the overall redox field energy within the cell. I want you to view the total redox energy within the cell as just that, a field, where the field strength everywhere is increased with the introduction of each minute kinetic energy influx. In this way, redox reactions within the cell, and this is important, occur sequentially as their threshold energy occurs. Now, do you see how the principle of least action works? It is the redox field strength that continually increases, and at each new field strength, the available energy is always the minimum required by the next cell synthesis step. Behavior is not without foundations in the literature. The Hungarian physicist and Nobel laureate Albert Szent-Györgyi proposed a mechanism like this field strength argument in the 1950s, when he likened energy movement in a cell to semiconductor behavior. He postulated that energy accumulated as field strength might then be localized at one location where it could perform useful work. But I want us to look deeper into this process to fully understand its consequences. Not only is the cell synthesis function, covalent bond formation, propelled forward by the increased redox field strength, but with it, the amino acid chains of enzyme molecules are systematically transformed to their condensed active form. Let me explain that for a minute. It was Jacques Monod who summarized the state of enzyme form and function in 1971. He wrote of their remarkable structural fidelity, their precise globular forms, and unerring specificity. These enzymes were the subject of his life's work. But in the end, he concluded that something beyond genetically determined peptide sequences imparted the information content found in their condensed form. It is, I believe, the increase in redox field strength that sponsors a precise sequence of minute conformational changes that causes the enzyme's peptide chain to fold into its precise globular form and acquire its unerring specificity. This might well be what Monod refers to as the ultima ratio. This very systematic folding of proteins has two effects on the physics of the cell interior. First, it enhances the negentropic character of the enzyme's amino acid chain. But secondly, it adds to the cell's redox field strength and it does so in a very global context. Do you see how increasing redox field strength affects every part of the cell's cytoplasm? The field is universal within the cell's membrane. Thus, conformational changes are summarized in this very systematized manner, but not just one protein at a time, but along a timeline that propels multiple separable processes, all of which contribute to the cell's overall least action synthesis process. But there's more. The cell's overall synthesis script includes many subscripts. These materialize as cell organelles are synthesized. Organelles have their own internal redox field, which is separated from the cell's overall redox field by its membrane. And that membrane is scripted with a very specific set of reaction sequences that accomplish a distinct cell function. For instance, the membrane might be specific to 1 or 3 electron reactions rather than the 2 electron reactions described for the cell membrane. Organelle-specific reaction sequences should operate in ways that are similar to those that I proposed earlier for the cell membrane. Evolution has endowed each of these organelles with a specific function, and it's interwoven into the cell's overall cytoplasmic function. All of the minute steps involved in this increasingly complicated cell synthesis function are occurring one step at a time and in exactly the same order that occurred when the cell was last in precisely this redox field strength. You have probably already imagined that the overall redox script has branches that arise because of intermediary products composed by other supporting subscripts. Finally, it is important that we make the connection between the cell's redox field strength and the heat theory which initiated much of this discussion. Because redox field strength is directly related to the radiation domain energy stored between covalent electrons, it is also a measure of radiation temperature T sub r. We are now ready to move our attention from synthesis into cell function. This isn't really a new subject. Function is merely a subset of the synthesis function where the molecular and energy transforms realize more complex outcomes. I have already discussed one of these, the cell organelles having their own specific transmembrane reaction sequences and also their own internal redox field. So let's begin our discussion of complex cell functions. Remember how the far from equilibrium blackbody spectra had only 2 degrees of freedom? The first is associated with the thermodynamic temperature, T sub m. It causes changes in the vertical direction, and it is responsible for increasing or decreasing the number of quanta stored at existing frequencies. It tells us how hot an object is. The radiation temperature, T sub r, on the other hand, moves the Wien frequency horizontally, resulting in additions to the spectral curve's high-energy quanta. This transformation represents increased redox field energy within the cell. You need to understand these 2 degrees of freedom because they are essential to understanding more advanced cell functions. I should also tell you that my goal here is not to present solutions for complex cell functions as I tried to do in discussing metabolic function. Rather, I want to outline for you some of the issues that need to be addressed and then describe the context within which complex cell functions might be studied. I want you to see the initial steps in the research process where hypotheses are proposed, questioned, and then modified. Let me begin by summarizing in a very broad sense how this model represents cell structure. The cell membrane encloses a cytoplasmic mass having a redox field strength that is always increasing. Within the cytoplasm, there are organelles having very specific functions. These include the mitochondria, ribosomes, the endoplasmic reticulum, and the nucleus itself. I want you to understand that organelle membranes separate regions having very different redox intensities in the same way that the cell membrane separates the aqueous environment from the cell's redox environment. Thus, the organelles are very localized regions of very different redox chemistry. Basic metabolic functions occur within the cell's cytoplasm, while more specialized, higher energy functions can occur within organelles. For example, our cell membrane had reaction sequences that are functionally two-electron reactions, and they form very high-energy intermediary hydrogen peroxide. It is possible that the cell's internal organelles might have very different reaction sequences, involving 1, 2, or 3 electron reactions. And it's likely reactions involving an energy intermediary other than hydrogen peroxide. Do you see how this model allows for entirely different redox chemistry across each internal membrane, and also entirely different redox field strengths within each membrane? This permits different functions occurring at different redox energies in different organelles. In fact, there is nothing that would preclude entirely different least action processes in different internal organelles. It would be interesting to work out a mechanism whereby the overall cytoplasmic redox field regulates the transport across other internal membranes. Are you beginning to see how the context provided by a theoretical model is suggesting questions to the theorist? Questions then lead to hypotheses that can be tested in both theory and experimental studies. To begin the hypothesis development process, we need to understand how metabolism and synthesis occupy well-defined regions in the far from equilibrium energy distribution diagram. Then, within the synthesis region, several functional subregions can be identified. These correspond to stages of the cell cycle. And because the redox field strength within the cell increases continually during the cell cycle, the subregions correspond to different redox energy states. The lowest of these is the energy region below G1, where basic metabolic function is localized. The highest energy regions are those that occur late in M phase. Let me also tell you something about cell division. Daughter cells have exactly half of the parent cell's molecular structure. This means that there is an equal or very close to equal split in the number of both covalent bonds and redox energy between the two daughter cells. And because the radiation temperature Tr is causally related to the total covalent bond energy and the redox field strength, Tr within the daughter cells is reduced to about half of the value that it had in the parent cells. If we now compare the difference between the cell's pre- and post-mitotic energy structure, we find that the difference between the parent cell and the daughter cell's energies is the synthesis potential that each daughter cell needs to create on its way to its next cell division. Remember, the process that brings about this is inevitable. And although there may be countless metabolic events occurring simultaneously, the synthesis path to the next cell division remains exact and very precise. Finally, I want you to recall that the entropic direction of energy decay is from the more organized radiation domain energy, think T sub r, into the domain of molecular motion represented by T sub m. I want you to see that within the cell, this would be a shift in the cell's redox energy from higher energy covalent bonds to lower energy covalent bonds, and consequently a shift in covalent bond energy in the direction of earlier stages of cell development. In other words, during aging, the high energy storage in mitotic phase covalent bonds might be systematically transferred to lower energy bonds in the nucleotide synthesis region of the far from equilibrium spectral curve. This shift favors the replication of pre-DNA proteins and nucleotides, DNA synthesis, aneuploidy, and a reduction in the high-energy covalent structures that occur later in the cell cycle. As this shift in energy occurs over many cell cycles, cell division becomes increasingly less likely. With this introduction, let's begin to look at the first complex cell function, aging. In its simplest explanation, aging might be viewed as a process where the cell simply loses energy. In our model, this could occur as a reduction in the number of covalent bonds, with its resulting loss in redox field energy. Or alternatively, energy might be shifted from higher energy covalent bonds to lower energy bonds. The energy difference would probably have to take place across an organelle boundary. But regardless of how we change the cell's energy structure, we must always be mindful of the first law's energy conservation principle. In any reversible process, we will have to demonstrate energy closure. A systematic reduction in the number of covalent bonds might, for example, occur if the original number of covalent bonds at a specific energy level is odd. An uneven split would give one daughter one fewer covalent bonds at all such frequencies. Alternatively, the bond efficiency might be split equally between the two, in which case both cells age equally. In an alternative scenario, telomere shortening could explain covalent bond loss. However, what I want you to notice is how in the division of odd numbers of covalent bonds, energy conservation is easily traced. While in the telomere shortening case, it is not immediately apparent how energy conservation occurs. Where does the telomere bond energy go to? You could probably come up with other mechanisms whereby both daughter cells have a lower radiation domain temperature. Let me suggest that the missing bonds are probably among the last formed during the cell cycle, the molecular components of, for example, the mitotic spindle. In any event, both daughter cells have a lower redox field intensity, and both cells have aged. A different type of energy loss scenario can occur if there is a systematic shift from higher energy covalent bonds to low energy bonds. But it is unlikely that this could occur within a single membrane. Within the context of this model, redox field strength is the same everywhere within a given membrane. Thus, two different redox fields can only exist across a cell membrane that delineates, for example, a cell organelle. Perhaps you have an idea that fits with this type of aging. The theorist's next step focuses on quantifying, for instance, the number of energy quanta lost in each cell division. In the example where the number of covalent bonds are odd and even, this task might involve the far from equilibrium blackbody equation, using it to first calculate if there are odd and even numbers of sites on the ascending and descending portions of the curve. Then the theorist might make estimates of the possible range of radiation temperatures in an animal cell, based perhaps on an estimate of the number of covalent bonds in the parent cell, and then calculate the total energy in regions where the number of quanta is an odd number. Do you see the process here? We are slowly developing a hypothesis. At this point, the working hypothesis for additional theoretical work might be either aging alters the cell's redox state by systematically reducing the number of covalent bonds in daughter cells following cell division, or aging alters the cell's redox state by lowering the cell's redox field intensity, possibly shifting covalent bond energy in the direction of earlier stages of cell development. Do you see the kind of questions that are involved in making the transition from theory to quantitative biology? You bring the theory's hypothesis to life by quantifying theoreticals and then testing those numbers against whatever observations are available, or simply asking, is this calculation reasonable? Does it conflict with anything that I know about the cell's aging? Eventually, the theory develops to the place where metabolic function resolved to, a place where you can begin to compare calculations to literature values and even formulate experiments to test elements of the theory. This process is important. Let me restate it. I'm using theoretical concepts to describe quantities that can be calculated. In essence, I am replacing ifs, ands, and buts in the original hypothesis with calculations based on probable ranges of model coefficients. This is the beginning of the hypothesis development process. You make repeated calculations, always checking that the numbers are reasonable, but more importantly, always checking that it doesn't conflict with what we know already, and even more important, always checking that it doesn't conflict with what has been observed experimentally. This is how the theorist uses quantitative biology to develop and refine hypotheses and ultimately formulate experimental tasks. Let me take us next into a brief discussion of natural selection. I want you to step back and look at our cell theory from an evolutionary standpoint. What we have theorized so far is a very highly organized sequence of synthesis steps that is the end product of natural selection. This is the cell's script. It is a very stable process. During the evolutionary process, small changes have occurred in the sequence of synthesis steps that produce gamete cells. These result in mutations in the cell line's molecular structure. It might be a change in the DNA that produces a slightly different protein that then alters the cell's least action process by, for example, inserting an extra synthesis step. In subsequent cell cycles, the cell's least action process incorporates that extra step and produces a slightly different molecular structure, and natural selection determines the long-term outcome. When multicellular organisms have these molecular changes or mutations in a subset of their cells, or perhaps even a single cell, a high-speed version of natural selection takes place. Instead of testing new traits over many generations of the organism, the new trait is tested in successive cell divisions. Cells having the modified least action process follow a new script. Mutated cells can die off immediately, or if it's changed to the cell's least action process is stable, its effects can be accumulated in the organism, but it cannot pass it on to its progeny. Cancer is a different kind of mutation. I believe that it is not in the cell's molecular makeup. Instead, I want you to consider the possibility that it is a mutation of the cell's energy structure. In the cancer phenotype, redox field energy is added to the cell, but there is no instruction set on how it is to be used. Consider the effect of chemicals having a high internal redox field strength, PCBs for example, or a virus with a redox energy storage higher than the cell's normal maximum, or a direct input of electromagnetic energy. In all of these circumstances, T sub M increases, and with it pre-DNA proteins and amino acids, while T sub R also increases, giving rise to increased mitotic potential. The cell's redox state is no longer consistent with its mass and radiation temperature states. The arbitrary influx of high-energy covalent bond potential, without a realistic template for how the energy is to be structured, leaves the cell with more nucleotide synthesis than it needs and an elevated post-mitotic redox state. It produces unneeded amino acids, nucleotides, DNA, mitotic molecules, etc. And because the redox field intensity is elevated beyond that required for mitosis, even in the new daughter cells, the cell cycle continues unabated. This is a disruptive change in cell cycle activity. The script is no longer organized by evolution, but rather by an arbitrary influx of covalent bond energy. The new synthesis path has no roots in the very organized evolutionary process that produced the initial normal cell. The extra energy is resolved by a sequence of least action steps that are without precedence and also without purpose. Do you see why cancer has eluded understanding? It is most fundamentally a disease of the cell's energy structure. Its cause cannot be seen in the cell's molecules. The altered cell chemistry is merely a product of that same aberrant energy structure. Until measurement methods are fine-tuned to this cause, cancer can only be understood in theory. However, I believe that this can change rapidly. By way of summary, let me reiterate the most important point that I have endeavored to bring to your attention in this lecture. It is an understanding that we only see about one half of the cell by limiting our experimental and theoretical efforts to molecular forms that we can see. An equally rich and largely unexplored understanding of the cell's form and function is to be found in its energy structure. A new theory of heat provides at least a starting point for initiating this kind of research. It allows us to imagine an energy scaffold upon which a model of molecular biology can be built. It also allows us to speculate on how aging occurs because of an entropic transition in the cell's energy structure, or how cancer is fundamentally a disease of that energy structure. This cell theory requires one element of new science, a theory of heat that brings together into a single equation the two existing theories bearing the revered names of Max Planck, Ludwig Boltzmann, and James Clerk Maxwell. This theory permits energy calculations for the type of very far from equilibrium energy storage that characterizes matter's living state. The model partitions all cell functions into two classes, metabolism and cell synthesis. Each of these can be modeled within a context that uses free energy relationships to describe cell functions. The results present plausible explanations for metabolic stasis, as well as the temporal progression of cell synthesis events. Reversible thermodynamics has a distinct advantage in modeling the cell's precision. All reversible processes operate strictly in accordance with the principle of least action, which means that the process is precise and absolutely deterministic. It is gratifying to model a living cell with this kind of exactitude. However, the implications of this research goes further. I've titled it Introduction to Theoretical Biology to emphasize its purpose in illustrating a productive approach to systematized biology. I want to emphasize that the kind of science illustrated here does not require a laboratory. Instead, it requires quiet time for exploring hidden corners of physics and chemistry and thinking about their implications in light of the vast observational record in the biological sciences. Each time that we do this exercise, we are taking a step toward context. And context is our single most important objective as we reinvent theoretical biology. Thank you.